Ecology 101 for the Praxis Biology 5236. Complete breakdown preparation quiz. Question 1. A physiology class is studying the human circulatory system. The teacher explains that blood flows through the heart in a specific pathway to ensure efficient oxygenation of blood throughout the body. A student asks why the heart has 4 chambers rather than just 2 like in fish. Which of the following explanations best describes the evolutionary advantage of the 4-chambered heart in mammals and birds? Is it? Option A. The 4-chambered heart allows complete separation of oxygenated and deoxygenated blood, which enables higher metabolic rates and endothermy, by delivering more oxygen to tissues than would be possible with mixing of blood. B. The 4-chambered heart provides redundancy so that if one chamber fails, the other 3 can continue pumping blood, increasing survival after heart attacks compared to animals with 2-chambered hearts. C. The additional chambers allow the heart to pump blood at higher pressure, which is necessary for animals that live on land, because gravity makes it harder to circulate blood compared to aquatic environments. Or D. The 4-chambered heart evolved to accommodate the larger body size of mammals and birds, as 2-chambered hearts cannot generate sufficient pressure to circulate blood through longer circulatory pathways. The correct answer is A. This question addresses the relationship between heart structure, metabolism, and thermoregulation. Fish have a 2-chambered heart, atrium and ventricle, that pumps deoxygenated blood to the gills, where it picks up oxygen, then flows directly to the body. This means some mixing of oxygenated and deoxygenated blood occurs, and blood pressure drops significantly after passing through the gill capillaries. Mammals and birds have a 4-chambered heart with 2 separate pumps. The right side pumps deoxygenated blood to the lungs, and the left side pumps oxygenated blood to the body. This complete separation ensures that oxygenated blood is delivered to tissues at high pressure, supporting the high metabolic rates required for endothermy, maintaining constant body temperature. Option A correctly identifies this advantage. Option B is incorrect because the 4-chambered structure does not provide redundancy. Failure of any chamber is life-threatening. Option C has some truth about pressure but oversimplifies. Many land animals with 3-chambered hearts, amphibians, most reptiles, manage gravity adequately. Option D is partially true but not the primary evolutionary driver. Some large animals have 3-chambered hearts. Complete separation of oxygenated and deoxygenated blood is the key adaptation supporting high metabolic rates. Question 2. A student is observing cells under a microscope and notices that some cells have visible chromosomes lined up along the equator of the cell. Other cells show chromosomes being pulled toward opposite poles. A third group of cells has 2 distinct nuclei forming. The student correctly identifies that these cells are undergoing mitosis. Which of the following sequences places these observed stages in the correct chronological order, as they would occur during mitosis? Is it? Option A. Chromosomes lined up at equator, chromosomes pulled to poles, 2 nuclei forming. B. Chromosomes pulled to poles, chromosomes lined up at equator, 2 nuclei forming. C. 2 nuclei forming, chromosomes lined up at equator, chromosomes pulled to poles. Or D. Chromosomes lined up at equator, 2 nuclei forming, chromosomes pulled to poles. The correct answer is A. This question tests knowledge of the stages of mitosis in correct sequence. Mitosis proceeds in a specific order. Prophase, chromosomes condense. Metaphase, chromosomes line up at the metaphase plate/equator. Anaphase, sister chromatids separate and are pulled to opposite poles. And telophase, nuclear membranes reform around 2 sets of chromosomes, forming 2 nuclei. The observations described correspond to metaphase, lined up at equator, anaphase, pulled to poles, and telophase, 2 nuclei forming. Therefore, the correct chronological sequence is metaphase, then anaphase, then telophase, which matches option A. Option B reverses metaphase and anaphase. Option C places telophase first, which is impossible as telophase occurs after chromosome separation. Option D places telophase before anaphase, which is also incorrect. Understanding the sequence of mitotic stages is fundamental to comprehending how cells divide and distribute genetic material equally to daughter cells. Remember, we can take your Praxis 5236 exam for you remotely and undetected with 160 score guaranteed. Message us via the number in the bio. Question 3. A biology student is studying the process of cellular respiration and creates a diagram showing the major stages: glycolysis, the Krebs cycle, and the electron transport chain. The student labels where ATP and ADH and FADH2 are produced. Which of the following correctly summarizes the net ATP production from one molecule of glucose through aerobic respiration in eukaryotic cells? Is it? Option A. 2 ATP from glycolysis, 2 ATP from Krebs cycle, and about 34 ATP from electron transport chain, for a total of approximately 38 ATP. B. 4 ATP from glycolysis, 2 ATP from Krebs cycle, and no ATP from electron transport chain, for a total of 6 ATP. C. 2 ATP from glycolysis, 2 ATP from Krebs cycle, and about 2 ATP from electron transport chain for a total of 6 ATP. Or D. 2 ATP from glycolysis, 32 ATP from Krebs cycle, and 4 ATP from electron transport chain, for a total of 38 ATP. The correct answer is A. This question tests knowledge of ATP yield in aerobic respiration. The accepted approximate ATP yield from complete oxidation of one glucose molecule in eukaryotes is: glycolysis produces 2 ATP net, after accounting for ATP used in investment phase, and 2 NADH. The NADH from glycolysis in eukaryotes yields about 1.5 to 2.5 ATP each depending on shuttle system, typically around 1.5 for the glycerol phosphate shuttle or 2.5 for the malate aspartate shuttle. The Krebs cycle produces 2 ATP, or GTP, directly 6 NADH, and 2 FADH2 per glucose. Each NADH yields about 2.5 ATP, each FADH2 about 1.5 ATP through the electron transport chain. The total is approximately 30 to 32 ATP from oxidative phosphorylation plus 4 from substrate-level phosphorylation, 2 glycolysis, 2 Krebs, giving 34 to 38 ATP total. Older textbooks often cite 38 ATP, while modern estimates are closer to 30 to 32 due to updated proton pumping stoichiometry. Option A gives the classic 38 ATP total, 2 plus 2 plus 34. Option B incorrectly states no ATP from ETC. Option C gives only 6 ATP, which is anaerobic yield. Option D incorrectly assigns most ATP to Krebs cycle directly. The correct answer reflects that most ATP comes from oxidative phosphorylation, with glycolysis and Krebs providing substrate-level ATP and reduced carriers for the ETC. Question 4. A student is examining a cross-section of a leaf under a microscope and observes numerous small openings on the lower epidermis, each flanked by 2 specialized cells. The student correctly identifies these as stomata with guard cells. The teacher asks the student to explain how these structures contribute to both photosynthesis and water balance in the plant. Which of the following explanations accurately integrates the functions of stomata with plant physiology? Is it? Option A. The guard cells contain chloroplasts and perform photosynthesis to produce glucose that is transported to other plant parts, while the stomatal opening allows oxygen needed for cellular respiration to enter the leaf. B. The stomata open when guard cells become turgid due to potassium ion uptake, allowing carbon dioxide to enter for photosynthesis, but this opening also results in water vapor loss through transpiration, creating a trade-off between gas exchange and water conservation. C. The stomata remain open continuously during daylight hours to maximize carbon dioxide uptake for photosynthesis, and any water loss is insignificant because the plant continuously absorbs water through its roots to compensate. Or D. The guard cells respond to light intensity by closing the stomata during peak sunlight hours to prevent excessive water loss, then opening them at night when carbon dioxide can be absorbed without water loss. The correct answer is B. This question tests understanding of stomatal function and the physiological trade-offs in plant adaptation. Stomata are critical structures that balance the plant's need for carbon dioxide uptake for photosynthesis against the inevitable water loss through transpiration. The correct explanation integrates the mechanism of stomatal opening, guard cell turgor changes driven by ion uptake, the benefit, CO2 entry for photosynthesis, and the cost, water vapor loss. Option A is partially correct about guard cells containing chloroplasts, but their photosynthesis primarily provides energy for guard cell function, not for export, and stomata do not primarily function for oxygen entry. Option C is incorrect because stomata do not remain continuously open. They respond to environmental conditions, and water loss can be significant enough to cause wilting if not regulated. Question 5. A biology class is studying the process of meiosis and its role in genetic variation. The teacher explains that 2 key mechanisms during meiosis generate genetic diversity in gametes. A student asks how these mechanisms differ from mitosis in their contribution to genetic variation. Which of the following explanations accurately describes the unique sources of genetic variation in meiosis? Is it? Option A. Crossing over during prophase I and independent assortment during metaphase I create new combinations of alleles on chromosomes and in gametes, while mitosis produces genetically identical daughter cells with no recombination. B. Mutation rates are higher during meiosis than during mitosis, which is the primary source of genetic variation, while mitosis has strict proofreading mechanisms that prevent almost all mutations. C. The random fertilization of an egg by a sperm is the only source of genetic variation in sexually reproducing organisms, and meiosis simply reduces chromosome number without contributing to diversity. Or D. Meiosis produces 4 daughter cells instead of 2, which automatically doubles the genetic variation compared to mitosis, regardless of crossing over or independent assortment. The correct answer is A. This question addresses the mechanisms of genetic variation in sexual reproduction. During meiosis I, 2 key events generate genetic diversity. Crossing over in prophase I involves exchange of genetic material between homologous chromosomes, creating new combinations of alleles on individual chromosomes. Independent assortment during metaphase I refers to the random alignment of homologous pairs, leading to different combinations of maternal and paternal chromosomes in gametes. These processes, combined with random fertilization, generate enormous genetic diversity. Mitosis produces genetically identical daughter cells and does not generate variation. Option A correctly identifies crossing over and independent assortment as the unique sources of variation in meiosis. Option B is incorrect. Mutation rates are not inherently higher in meiosis, and mutation is not the primary source of meiotic variation. Option C is incorrect because random fertilization contributes to variation, but is not the only source. Meiosis itself generates variation. Option D is incorrect. Producing 4 cells does not inherently create genetic variation. They could be identical without crossing over and independent assortment. The correct answer distinguishes the unique recombination mechanisms of meiosis. How many did you score out of 5? Let us know in the comments below. Also, tell us which exam we should cover next. If you found this video helpful, please like, share, and subscribe for more.